Hi everyone, and welcome to week 8 of lab. We have a few different tasks for you this week. First of all, we would like you to sign up for a time to conference with your lab instructor. The purpose of these conferences is to talk about the process of writing your yeast manuscript. What went well, what was challenging, and you'll also get a chance to discuss the feedback we'll give you on your original draft as you start working on your rewrite. Take a look at the links on this post for more instructions on how to sign up for a conference time. This week, you will also get started on a multi-week restriction mapping experiment, which will be the subject of your final research manuscript. In this experiment, you are going to use a technique called restriction mapping to differentiate between two plasmids. As you learned in Bio 101, plasmids are circular molecules of DNA found in bacteria, separate from the bacterial chromosome. They contain one or more genes, and when a bacterium takes up or is transformed by a plasmid, it will start expressing the genes carried on that plasmid. In molecular biology, plasmids are often used as cloning vectors. Molecular biologists can take a piece of DNA that they're interested in, insert it into a cloning vector plasmid, and then transform, say, E. coli with that plasmid. By growing up a lot of this transformed E. coli, you can then get a lot of copies of your interesting gene. In addition to the gene of interest, cloning vector plasmids will often have a gene that functions as a selectable marker. This marker gene gives you a way to tell whether bacteria has been successfully transformed or not. Often, it will be something like an antibiotic resistance gene, so that you can grow your bacteria culture on a plate that contains that antibiotic, and you'll know that all the bacteria that survive on that plate must have the plasmid. In order to insert a gene into a cloning vector, molecular biologists use restriction enzymes. A restriction enzyme is an enzyme that recognizes a certain sequence of nucleotides in a piece of DNA, and when it finds that sequence, it makes a cut. Digesting a plasmid with a restriction enzyme that cuts just once will open it up, allowing you to insert the gene of interest. Because restriction enzymes recognize specific sequences, different plasmids will be cut in different places when you expose them to the same restriction enzyme. This means you'll get different numbers and sizes of fragments depending on the sequence of the plasmid you digest. By digesting a plasmid with two different restriction enzymes, first separately and then together, it's possible to make a map of the relative locations of restriction enzyme cut sites on that plasmid. This process is called restriction mapping. In your experiment, you're going to use that logic to identify which of two possible plasmids you have in your sample. The DNA sequences of these plasmids are known and have been given to you. You're going to import them into the program SnapGene, which can analyze the DNA sequence and show you where on those plasmids different restriction enzymes cut. This will allow you to choose a pair of restriction enzymes that will cut the two possible plasmids into unique numbers and sizes of fragments, allowing you to differentiate between them. In the coming weeks, we'll virtually go through the steps of transforming E. coli, isolating plasmid, performing the restriction digests you chose, and running the resulting fragments on an agarose gel to determine their sizes. In the end, you will get results from which you'll be able to identify your unknown plasmid. The third and final task for this week is to start on the data analysis from the Drosophila experiment. At this point, you have the F1 data from your group, and the F2 data from the class has been pooled and posted for you. Based on your observations, you should be able to make hypotheses about how the W, B, and VG genes are inherited. To test these hypotheses, you're going to use chi-square tests. The process will be similar to the chi-square test you did during the Sordaria lab. Based on your knowledge of genetics, you should be able to predict the phenotypic ratios you would expect in the F2 generation if your hypothesis about a given gene is true. You can then use a chi-square test to compare the expected number of, say, white-eyed flies and red-eyed flies to the number of white-eyed and red-eyed flies you actually observed. The exercise walks you through the logic of the analysis step by step. If you run into trouble with anything pertaining to this week's lab, remember that you can post questions on the relevant discussion boards on Canvas.